And congratulations on the amazing panel discussion we just had inside. It was a very insightful discussion for us and for all our audience. So I would like to start. What is your experience, your views about this event, and how do you think this can pave future?
Thank you for that compliment. I think this was a one of a kind conclave, scientific conclave that organized by HORIBA. I think where there was really truly transparent discussion between stakeholders, industry, academia, and multinational companies. So really very good exchange of ideas wherein scientists presented their little some work that they have done, but more importantly industry presented emerging technologies that are already making a difference and will have a bigger impact in various aspects in industries such as biologics, but also in healthcare. So I think it was a unique combination of experts from various disciplines bringing ideas and future trends in healthcare. Yeah.
That's great. So moving on to my next question, which is a follow-up question to the panel discussion we had.
Yes.
Can you discuss the role of advanced biological techniques in unraveling complex cellular mechanism and their potential impact on therapeutic product developments?
This topic is close to my heart actually, so I'm happy you asked me this question. Advanced technologies will always be necessary, and I can give you some examples. I'm a great fan of technologies in terms of not only do I use them, but also I develop technologies and take them to where it's needed the most. So I'll give you an example of mass spectrometry, which is a technology which actually appeared very esoteric, very limited to highly academic labs because of the cost of the equipment, at the same time understanding how to use that technology, and that's typical with most technologies. But in case of mass spectrometry, over the last 2 decades, what I've seen, and I've been a part of this whole process of getting the technology across, is that now industries have adopted this as a routine part of their process of characterizing biological therapeutics. And that's the question you had, the therapeutic molecules, development of therapeutic molecules. So it's become an integral part, and I often use this analogy. It's really become like a Swiss knife of a biosimilar industry. It actually does multiple so-called critical quality attribute analysis in a way that otherwise you would need 10 other instruments to do, and in a limited time with very limited. So this is a wonderful example that in biologics, in biological biopharma product characterization role that mass spectrometry has done, and I have played a very important role in this, in educating industry, educating the regulators, and also training people in the industry to be able to use this technology efficiently. The other technology that we heard about today, I think, is going to make a significant impact, and I'm very thrilled about it because I think it's a technology that was developed by an Indian, and that's we all know what we are talking about, Raman Spectroscopy, right? I'm so proud to be associated with an institution where Sir C.V. Raman was one of the first directors, but also the only Nobel Prize winner from that we had close association with as an institute. I think this technology of using Raman spectra to characterize wide variety of molecules, I think is going to really revolutionize once this technology becomes available in an easy way. Currently it's used by industry because the instruments are very expensive. What's going to happen, and I hope it will happen, and I'm myself very keenly observing this, is it's going to become user friendly, and in a way that I think the user may not even know what Raman spectroscopy is, what's Raman spectra, yet they are able to get solution to problems that they are addressing. And that's when we know that technology is successful, which is the case with many things that we have seen over the years, right? I think today, I think that's a small answer to the question that you have. Yeah.
That's great, sir. You answered very well, and thank you so much for sharing your insights with us, and thank you so much for giving your time and coming to the event.
Such a pleasure. Thank you so very much. Thank you.